very interested in the role of RNA in early evolution of life. And today I will tell you a little bit about our experiments that we've done in that area. Specifically about our interest in determining the relative role of different evolutionary mechanisms in the evolution of functional RNA. And part of the motivation behind that is the fact that in biology there are a number of different evolutionary mechanisms that operate at creating diversity and creating more functional polymers or forms. And of those, only a number have been investigated by in vitro evolution studies. Primarily, the dominant mechanism that has been investigated is point mutation. And that is partly driven by the fact that a number of theoretical studies have suggested that RNAs of increased complexity or increased length are connected by large swaths of neutral networks, which would allow them to, by point mutation, traverse large swaths of sequence space, therefore give them the ability to potentially change structures in very radical ways. So the way we studied this is by evolution of ligase ribozymes, and we evolved two separate independent ligase ribozymes, one short one of 20 nucleotides and one long one of 80 nucleotides. And a typical in vitro evolution experiment begins with a diverse library. In this case, this is a 20 nucleotide random region that's flanked by two constant regions, and we transcribe this into RNA and then incubate with the substrate to allow for ligation. In this case, the substrate brings about it is a constant region, after that, the partitioning reaction separates the active molecules from the inactive ones. In the case of the first round, that of course is the number of the inactive molecules dominates, and then the small number of active ones is then reverse transcribed, PCR amplified, and then converted back to DNA. The process continues. We did this for the 20 nucleotide selection, and then for the 80 nucleotide selection, in which case it was 8 rounds of selection punctuated by mutagenesis that was after the 5th round. So what do we get? The winners of the 20th selection are these ligases. They have the ligation junction that's not base paired, and this stem loop structure. The dominant molecules from the 20th selection are, how do we count them? Basically, we look at their abundance in the final population, and we can do this because there is the representation of the molecules in the initial pool is very large, so we have high copy number in the starting pool. And so what do these populations look like? This is my way of representing them. So we clustered them into sequence networks based on their ability to connect through point mutation. And the final population looks the way I imagine it, something like this. So we have two dominant networks. One is very small in the number of sequences, but they're very abundant. So those are the winners, the ones we expect to be very active. And the larger pool of sequences that represent the broader swath of sequence space, but are very low in abundance, so we don't expect them to be very active. The ADN population, these are the winners from the ADN population. Of course, their ADN sequence space is very large, astronomically large, and so we have very sparsely sampled sequence space. What happens, so the winners are the most enriched sequences. They're not the most abundant ones, but the ones, they actually happen to be also the very abundant ones, but they are the ones that increase in abundance over different rounds of selection the most. What is the important thing here is that from these two unrelated selections, we get ligases that are the winners in their respective categories that are very similar in sequence or identical in sequence. So what we get is the same identical ligation junction, and then that the smaller motif is actually embedded within the larger motif, which suggests that the addition of this top stem loop structure could lead to evolution of higher activity ligases. So we wanted to test this, and we tested their activity. So the 20N ligase is not very fast, but the addition of the stem loop adds about gives a 1,000-fold improvement in activity. Conversely to that, the removal of the loop removes about 50-fold activity. And there's a trend. There's a log linear trend in activity versus length motif. So we wanted to test the extent of this. To do this, we took two very well-known ligase ribozymes, the L1 and class 2 ligase ribosome. They were old and have been optimized over time. And we placed them in the identical structural context as the ligases that we evolved. And what we find is that the trend still continues. We also did a bit of a literature search, and from the literature search, we find that there is this activity length trend among both aptamers, so for all functional RNAs, so aptamers such as GTP aptamer, and also other ligases. So we have here just a survey of different ligase activities that have been published. We wanted to examine the extent of this trend. So if we continue to grow these ligases, can we obtain even higher levels of activity? And to do this, we reselected the most active ligase, so the winner, the internal loop ligase. We mutagenized it in 40 positions for one of the trajectories, and then added a fully at 20% per position, so that's quite high in mutagenesis, and then added 20 fully random nucleotides to the top of the loop, and then for the third trajectory added 20 fully random sequence to the 3' end. So what do we get? What we find is that there is a limit to this trend. So what we get is 10-fold improvement in activity upon this internal loop motif, and the way that is achieved is by creating a slightly larger loop. We don't know the details of this mechanism, but that is what we observed. We also get a very similar activity for the same motif placed in a larger context. So from the third of the reselections, we obtained this motif, which is the exact same motif, just containing additional sequence element, and that has identical activity, but only slightly higher amplitude in activity. So the fraction of the molecules that ligate is just slightly higher. What is striking among these selections is that what we get is that the structure of the core motif does not change. So only the peripheral elements change, slight remodeling there at the top of the loop, but the core sequence remains completely unchanged, and the addition and then the remodeling of the top stem loop results in 1,000, 5,000, and then 10,000-fold improvement in activity. So the take-home message from this, and I kind of went through fast. So I didn't talk much about the sequence networks and their connectedness, but what we find is that contrary to the theoretical work that has been proposed, the sequence networks are disconnected at all different lengths. So for the short ribozymes, they're definitely disconnected. But what was slightly surprising was that for the reselection of the longer ones, we definitely see even smaller networks in sequence space. So the point mutation leads to only limited optimization potential, so it's very local optimization. Sequence insertions, so these large type of sequence insertions like recombination could lead to initially large improvements in activity, but then sort of start to lag. And the most important observation here is that the sequence insertions preserve the core structure, which is important for interpreting the molecular record today. So you can imagine if we look at modern structures today, to envisage what the earlier functional structures would have been. I'd like to thank people that have worked on this, current and past members of the Ditzler group, Teresa and Alex in particular, our collaborators, Andrew and Chenyu. I'd like to thank our funding sources, and I'd like to thank you for your time.
Thank you, Milena. We have time for some questions. It looks like if we'll start with Anton in the back.
If you look for sequence specificity to see if you have any some sequences with promote expansions in apical loops and three-way junctions.
No. Thank you, Anton. That's a great question. We haven't, but we are planning to follow up on this project with additional selections and looking more into detail about the types of sequences and structures that would promote sequence insertions. Because we do think, like your work, that sequence insertions was an important mechanism for increasing function early on. And that's something that particularly is important for RNA. It could have led to RNA, could be the underlying reason that RNA was and is an important molecule today.
We'll go to Donald.
Yeah, great. So that was cool. That was really nice. I like the idea about building up better function for a particular activity by getting more. On the biology side, we often go the other direction. Think about you've got this functional molecule now. What might it have done historically? What's its core? Ribosome, for example. And then start to say, well, this little tiny thing might have been more promiscuous to other things. Do you have any way to get at looking at whether your ligase molecule, your minimal core, might be more able to do something, some sort of other chemistry more promiscuously than the more elaborate species, which is better and better and better for a particular reaction?
I mean, I can imagine reselections for...
not to get better, screening, and I mean, what would you look at? What kind of reaction would you look at? I mean, other sequences, sure, but are there other chemistries that, I mean, out of the blue question there.
No, I mean, there's so many important reactions that would have been important prebiotically, right? So replication type of reactions important for metabolism. I'm not actually quite sure that I understand your question exactly, so maybe we can... Okay.
Kindnesses are always good to select for, right, Donald? Okay, we'll go to Phil.
Phil Dunlop. I have a question about the activity versus the length. And so you can get longer by just throwing on more nucleotides. And so sometimes people have quantified that with information content, looking at the number of bits of information through Shannon entities. And you have the reference up there from Gruder's work from the Shostak lab. And so I'm wondering if you tried to translate that length increase, usually have to do random mutations and so on, into some kind of information content of the increase in size.
Right. Thank you for that question. It's a good question. Right, so I didn't explain this fully, but the motif length that we talked about can be directly translated into the informational content. So this is not the exact length of the molecule, it is the motif length. So, yeah.
All right. So with that, I think thank Milena again and move on to the next speaker.